let us talk about eukaryotes. So now let us see how is DNA helix packed inside nucleus in case of eukaryotes. Children, now in prokaryote it is quite simple. So what happens in eukaryotes? In eukaryotes there are some special proteins which are present and they are positively charged and they are basic in nature, right? And these proteins are known as histone proteins. और ये जो proteins होते हैं ये क्या कहलाते हैं? ये histone proteins कहलाते हैं, right? They are positively charged base protein. ये जो positively charged basic proteins होते हैं ये histone proteins कहलाते हैं. And these proteins are rich in amino acid. And these amino acids are basic in nature. That is why they are basic. Now, as I mentioned before also that DNA is negatively charged and due to the presence of negatively charged phosphate group, right? And these proteins are positively charged. So obviously there always being an affinity between DNA and these positively charged proteins. Now due to this affinity, let us see what happens. DNA will wrap around the histone octamer. What is octamer? Octamer क्या होता है? Octamer यहाँ पे octa word आता है. So that means 8. Now what happen is this histone protein they form nucleosomes. They are very small proteins और 8 protein का group होता है. They together form histone octamer. Histone के 8 piece they collect together, something like that. This is 1, 2, 3, 4, 5, 6, 7 and 8. So 8 histone together they are known as histone octamer. Now children, what happened? DNA wrap around this histone octamer to form the structure और इसके चारों ओर DNA is wrap around, right? तो ये जो structure बनता है ये क्या कहलाता है? Nucleosome. So, as similar with this structure, right? So this is how the nucleosome will look like. So this pink color is the histone protein and this blue color thread are the DNA, right? So in that way DNA attract with the positively charged proteins. So the structure which is formed, it is known as nucleosome, right? So here in this picture the pink color string which you see is the double stranded DNA. And here you can see a ball-like structure. That ball-like structure is the histone octamer and the DNA will wrap around the octamer to form the structure of nucleosome, right? So this is the structure of nucleosome. How is the structure formed? The structure is formed because the octamer was positively charged and DNA was negatively charged. So they got attracted towards each other and that DNA wrapped around the octamer. Okay, so now each nucleosome contain 200 base pairs of DNA helix. एक न्यूक्लियोसोम की अगर हम बात करते हैं तो एक न्यूक्लियोसोम को जो DNA रैप अराउंड करता है उसमें 200 बेस पेयर का DNA यूज होता है. So now you see that how it is going to DNA wrapping around the entire octamer, right? In one octamer the entire DNA which is present, it contains 200 base pairs. I was telling you before that one turn of the helix contains 10 base pairs, right? So when you say 200 base pairs, that means just 20 turns of helix. Clear? एक न्यूक्लियोसोम में 200 बेस पेयर यूज हो रहे हैं और मैंने आपको कहा था कि एक टर्न में 10 बेस पेयर होते हैं, तो तकरीबन 20 टर्न्स लिए होंगे डीएनए ने इस ऑक्टेमर को चारों तरफ, राइट? So that is how there will be some 20 turns of DNA which will be present in each nucleosome, right? Now children, when you look at a broader level, how does it look like? The entire structure which you see here, it looks very big, right? अब यहाँ पे आप ये जो nucleosome देख रहे हो देखने में तो बहुत बड़ा लग रहा है, but actually when you see, it is very tiny structure. When you observe it under an electron microscope, which is quite powerful microscope, how do they appear? So children, they will appear like beads on a string. ये कुछ ऐसा दिखेगा. They will appear as small, as tiny as beads. So this is how it look like. So normally this is a chromatin, look like the thread with the beads. Now if you observe it under the electron microscope, so each red color structure which you see, which appear like a bead, it is nothing but the nucleosome. इसमें जो आपको ऊपर ये दिख रहे हैं रेड कलर के जो बीड्स दिख रहे हैं, ये एक्चुअली न्यूक्लियोसोम को रिप्रेजेंट करते हैं, राइट. एंड जो इसमें आपको ये ऑरेंज कलर के जो दिख रहे हैं, ये आपको बताते हैं स्फेरिकल स्ट्रक्चर, दैट इज द हिस्टोन ऑक्टेमर, और दैट हिस्टोन ऑक्टेमर इज बीइंग रैप्ड अराउंड बाय द डीएनए, राइट. व्हाट इज दैट थ्रेड? सो this is nothing but the chromatin. In between you have nucleosomes. So this is how DNA is packed inside the nucleus. Now if you imagine it yourself, if DNA is wrapped over something, what has happened? It is like a thread, long chain. You're wrapping a chain over something. What is happening? You actually decreasing the length. समझ में आया? अगर किसी चीज़ के चारों तरफ आप किसी थ्रेड को रैप कर रहे हो तो क्या कर रहे हो? उसकी लेंथ को कम कर रहे हो. तो उसी तरीके से ये जो बीड्स हैं, इसके चारों ओर डीएनए लिपटा हुआ होता है, रैप किया हुआ होता है. तो एक्चुअली डीएनए की जो लेंथ होती है वो डिक्रीज हो रही होती है. व्हेन यू रैप इट ओवर द nucleosome, I mean over so many octamers, what happens? The length reduces a lot. It becomes so compact that it fits inside the nucleus. Got it? So this is the strategy of packing of DNA inside the nucleus. So one nucleosome contains 200 base pairs, and I already told you total base pair in typical mammalian cell is around 6.6 into 10 to power 9. So what would be the total number of nucleosome in a mammalian cell? तो टोटल नंबर ऑफ न्यूक्लियोसोम कितने होंगे? अगर टोटल नंबर ऑफ न्यूक्लियोसोम हम निकालें तो 6.6 इंटू 10 की पावर 9 डिवाइडेड बाय 200, क्योंकि एक न्यूक्लियोसोम में 200 बेस पेयर है. सो इट इज इक्वल्स टू 3.3 इंटू 10 की पावर -6. So these many nucleosomes will be present in a cell, and each of these nucleosomes will appear as bead, and that is why it often known as bead on a string, because chromatin appear as a string and nucleosome appear as bead, right? So the chromatin later during the cell division they condense together and form the chromosomes. So now that we have discussed about the chromatin, now let us talk about few more important points, something about the chromatin. So children, there are two forms of chromatin. One is euchromatin and the other one is heterochromatin. Now why I am sudden discussing about the forms of chromatin? The coiling and the condensing of the chromatin actually forms the chromosomes, and you all know that how much chromosomes important for the process of inheritance, right? So now here we will see that during this condensing, I mean when the condensation takes place into the chromosomes, these nucleosomes, they are not present everywhere. Somewhere they are more and somewhere they are less. So that is why we are discussing about euchromatin and heterochromatin. अगर euchromatin की अगर हम बात करते हैं तो DNA पैकेज जो होती है, it is looser. DNA is packed but it is not very compactly, or it is not very densely packed. There is a lot of empty spaces. So this region is known as euchromatin. It is loose packaging of DNA and light stained when studied under microscope, right? So this euchromatin, since it is less amount of DNA compared with the heterochromatin, because they are lightly stained, right? So when we study under the microscope, they are lightly stained areas, right? But they contain less DNA, transcriptionally active. So what do we mean by transcriptionally active? What happened is that for transcription, what is transcription? First of all, transcription is nothing but the formation of RNA from DNA. So for transcription, that portion of gene needs to interact with protein. In this structure, the region is accessible. So those regions are accessible. That is why that is transcriptionally active. वहीं अगर हम बात करते हैं हेटेरोक्रोमेटिन की, तो DNA is packaged very tightly. Lot of DNA is present there. More DNA, so it is darkly stained under the microscope. and it is transcriptionally inactive. That means that portion of DNA which need to exposed in order to synthesize RNA, that is hidden here, right? So what happened if you have too much of DNA? So what happens? It is very much compact, the portion of DNA which gene to be exposed for transcription, that does not happen. So that is why it is transcriptionally inactive. So appropriate region needs for transcription, they are accessible, and therefore transcription is inhibited. Therefore it is said that heterochromatin are transcriptionally inactive, but euchromatin is transcriptionally active. Okay, but when you talk about DNA content, heterochromatin has more DNA and euchromatin has lesser DNA. ठीक है, and euchromatin found in both prokaryotic as well as eukaryotic. और heterochromatin की अगर हम बात करते हैं, so they only found in eukaryotic, right? In prokaryotic, heterochromatin is absent. So this is what about the difference of chromatin. Now these chromatin condense to form chromosome during cell division. Now again during condensation of chromatin, they form chromosomes. So who helps? Again some proteins helps, and those proteins are known as non-histone proteins, right? So protein which help in DNA to pack itself, that is called histone protein, and the protein which helps chromatin to condense for further formation of chromosome, that is non-histone protein. So I hope that you get each and every point very clear. Children, still you feel some doubt, some queries, then just feel free to ask, right? So in the next video we will discuss further important things of this chapter. Till then, have a nice day and thank you.